change that. I think we can put, okay.
Okay, so welcome to the PhD defense for Tissot. So Ali approached me in 2018 about joining my lab as a master's student. After several years of trying to avoid following her father, the marine biologist's footsteps, as many kids do, not wanting to do exactly what your parents do. But she was ready to sort of embrace that action. So she talked to me about doing a PhD. Those two. Or I'm sorry, about doing a master's. And so I accepted her into our lab as a master's student to start in 2018. But I that I've actually on before. So I have been on committee with her dad's graduate students in the past. And even before that, I met Ali at the Western Society of Naturalists meeting when she was a high school student tagging along with her dad to the conference. This for at 15, which is the same with my daughter right now. And this isn't at the conference, but just so you can see where she came from attending WSI meetings. Next slide. Oh, sorry, wait, go back, sorry. Oh yeah, yes, sorry.
Oh, my arrows are all
Okay, sorry.
Yeah.
And then also re-meeting her when she was a undergrad, and because I did her own research at WSM. So I knew when she arrived at PSU that she had some background, and she had recently come off of working for Sergio Navarrete, who is what we might know, a well-known marine ecologist in Chile, doing fieldwork and lab work. So she had quite a bit of experience coming into the lab. But in many ways, she was also green because she had never designed her own full experiment and conducted it, and would have to ultimately be in charge of doing everything herself during COVID. And her, so her, she really had to hit the ground running, especially with COVID restrictions, where she could be in the building by herself in the lab at certain times when no one else was in the building, right? There were very strict rules. We had issues getting supplies for her experiment. And yet she conducted a really nice experiment we'll hear about soon, due to her tenacity, her positivity, and her ability to problem solve. But again, you'll hear more about her research a little later. So I just want to cover a couple of things that you may not know about Ali. So one is that you might know that she loves invertebrates, but you might not know that she has been interacting with and interested in invertebrates since she was a very young child, tagging along with her dad, right, so in the field. You also might not know, or you know, that after conducting that first chapter of her dissertation, which ended up being the very first chapter, but you'll see the progression, she came to me and said, despite having to do a lot of work by herself, struggling with getting materials for the experiments, having some great help from Steve, one of her community members, to collect clams in the field during COVID and be distanced from each other, etc. She came to me after this experiment and said, oh my gosh, that was so fun designing my own study and conducting my own study. Like, I have so many more questions I want to ask. Can I stay in your PhD? And so of course I said yes. And so another thing you might not know about Ali is that she's really been the super glue that's held our lab together for the past 6 years. She has taught new students and new people in the lab the ropes. She took on the role of safety coordinator for our lab. She's also made roles organizing an inclusive perspective on contaminant solutions, a symposium at the annual meeting of the Society for Environmental Toxicology and Chemistry. She filled in last summer when our PhD student wasn't able to get a visa to come before classes started, and she filled in as sort of a tech GRA for me on a microplastics project, which is a little bit further afield. And so she's just really been this key glue in our lab. Oh, sorry, you've already moved. Okay, go ahead.
Oh, okay.
Although Ali is going to be really sorely missed when she graduates, I'm also really proud to introduce another PhD student that is ready to fledge from the lab. And not really in her marine ecologist father's footsteps. She's actually really forged her own path as a marine ecotoxicologist. And so I'm really excited to see where she goes with that. And so now it's time for you to hear directly from Ali about her a bit more.
That's not fair. Gonna make me cry before
Worry about that.
I appreciate it. Okay, so let's see. I want to make sure. I'm probably gonna try to stay right here so that folks on Zoom can see me. So hopefully, can everybody hear me okay at this volume? Sweet. Okay. What we're also gonna do is. Does anybody know if we can hide this guy? I'm like, oh, bless. I love you. Okay. There we go. Okay. Nope. Nope. You're not supposed to see that yet. Go back. Okay. So we're gonna do captions. Oh, is that gonna pop up every time? Oh, yay. Okay. So hopefully the captions are helpful. They might be a little silly, but we'll see how that goes. Okay. All right. Thank you all so much. This is like so overwhelming in the best way. I really appreciate everybody coming and taking the time for this. Like Elise mentioned, this is 6 years in the making, so this is such a special moment. I really appreciate it. Welcome to my defense. Oh, you can't even see the thing. There we go. Okay. Welcome to my defense. So my title for my dissertation work is Pesticides as Multiple Stressors to Bivalves and Echinoderms. And I want to just take a second to really acknowledge my wonderful committee who's listed here on the left, especially my advisor, Dr. Elise Granick. So first, I want to talk about Rachel Carson. So for folks who don't know about her, she was a marine biologist turned toxicologist because she was learning about all these pesticides and how awful they were. And she kind of was a pioneer for environmental toxicology. So she's been a personal hero of mine for a long time. She wrote the book Silent Spring, which was really exposing pesticides for their toxicity in the environment and all the damage they cause. And so I really wanted to share this quote because I feel like it frames my research really well. That says, if we are going to live so intimately with these chemicals, eating and drinking them, taking them into the very marrow of our bones, we had better know something about their nature and their power. I love that quote. So a lot of her stuff was looking with terrestrial organisms. So we're going to transition to my dissertation topic, which is looking at what happens with all of these contaminants when they're exposed, aquatic organisms are so exposed to them. So, wondering if yeah, we'll let that go. So this is the agenda for today. I'm going to start out with an introduction, and then these are each of my chapters. So the first one is a background that talks about all of the different pesticides that I focused on. The second one is a field study that we got to do. The third one, yes. So yeah, the third one was the clam study that I got to do at the very beginning during COVID. And the final one is this toxicity study that I did with the larvae of the sunflower star, which was very special. So getting started with chapter 1, this is actually a picture from back when I lived and worked in Chile. We saw this washed up on a beach. It is one of those jugs that they keep pesticides in, and so it's breaking apart on the beach, and it says Monsanto on it. And I thought it was wild at the time, but now truly, given my full trajectory, I thought it was pretty powerful, so I decided to put that in there. So what are pesticides? They are essentially used to eradicate numerous pests. The main categories include weeds, rodents, insects, and fungi. They're widely used across sectors, so agriculture, forestry, and residential in people's homes. An example that I figured I would share that people might be familiar with is DDT, which is what Rachel Carson wrote about. This is an image from 1945 where they used to just spray it everywhere. This is on the beach, and I love this photo. So it has since been banned in the US because it is so toxic and very persistent in the environment, so it stays for a very long time. So there are a number of other anthropogenic contaminants in the environment, one of which is pharmaceutical and personal care products. So these are medications, and then also just like lotions, soaps, all the things that we use to take care of ourselves. Microplastics, which are defined as particles that are less than 5 millimeters in length. And then some other examples that I'm not going to get into in this presentation, but just so you can hear about them. So per and polyfluoroalkyl substances, which are PFAS, you may have heard of forever chemicals, or flame retardants, mercury. I have a picture of myself here because I'm wearing clothing that's shedding microfibers. I'm wearing rubber gloves that are probably shedding microplastics. And I also use pharmaceuticals and personal care products. So I myself am a vector for contaminants. So how do these chemicals get to the environment? There's a number of ways, but just kind of a quick overview. So some of them include runoff, leaching, and erosion, which are runoff is with water from the land to the aquatic environment. Leaching is through soil, and then erosion is essentially when a lot of soil moves from the land to the water. And then drift is when a pesticide can lift and vaporize into a cloud and move that way. Bioaccumulation, which is essentially where these contaminants are taken up into the tissues of an organism. And then biomagnification is where another organism consumes a ton of those organisms and takes up all of those concentrations, so it's much higher in that predator. So these are usually present at lower concentrations in the environment. However, they may produce a lot of non-lethal effects instead of just toxic effects, which are also important. So these include such things as reproductive effects, growth, and behavior. And as we're talking about all these contaminants in the environment, there's sort of a cocktail going on, but we don't really know what the effects of all of them are together. Additionally, we have all sorts of other stressors going on in the environment. So with climate change, we all know like temperatures increasing, which is really hard on a lot of animals, including humans. Increased runoff, there's going to be more rain or less rain, and so that's going to increase the movement into aquatic systems. And then very sadly, increase in disease frequency as climate change moves forward. So this is a wasting sea star, which we'll talk about again in a bit. So my first chapter, or second chapter here. So toxicity and environmental fate of atrazine, bifenthrin, diuron, indaziflam, and imidacloprid in aquatic ecosystems. So again, these are commonly detected in the Pacific Northwest. That's why they were chosen for my dissertation. First one, atrazine, they all have a lot of uses, but these are kind of the primary ones. So atrazine is used in agriculture and roadways. Diuron is primarily in aquaculture and antimicrobial, so it's actually in paint and stuff for like exterior of houses. And indaziflam is really commonly used in forestry and then also roadways for weeds and ditches. Bifenthrin is common in home insect management. Remember, my aunt actually showed me a bottle that they were using for ants, and it had bifenthrin in it. And imidacloprid is commonly used in agriculture and as a pet flea medication. So there are a lot of factors that influence the fate and transport, but some of the key ones include the sector of use. So where that is and how often, how close to a body of water. So this might include residential or agricultural, for an example. Application method. So this is seed coating. Sometimes they use this for pesticides that they don't want to move through spray or what have you. So you can see these are corn seeds that are covered in like a green coating. Backpack sprays are really common. And then helicopter sprays. And so you'll see each of these kind of have different potential for mobility through the environment. And then also just the physical and chemical properties of the pesticide itself. So often like how able it is to degrade. So like hydrolysis is breakdown through water. Photodegradation is breakdown with sun exposure. So for this, the background information, we're going to talk about the sectors of use, mode of action, which is essentially the specific toxicity target these chemicals are designed to have. And then their transport through the environment, which includes the route of movement, degradation, and then bioaccumulation, which again is their ability to be taken up by organisms. And then their toxicity, and then also non-lethal effects. So this is kind of just a big table. They don't have to take it all in, but in general, this is showing 3 of them are herbicides. So they're targeting plants, and then 2 of them are insecticides. And their general mode of action is just interesting to keep in mind. It shows you what it's going to do to its target organism. So the first two are photosynthesis inhibitors, kind of in a broad sense. They get a lot more specific, but we're not going to get too into today. And so yeah, and Dazaflam affects cell wall development, and then we've got nervous system effects with bifenthrin and imidacloprid. And then we've got all these uses. Agriculture, turf is really common, and that means golf courses and people's yards. So with this, their movement through the environment, we'll just take a second to kind of orient ourselves with this table. Down below, we've got our symbols showing the route, and then degradation and bioaccumulation factor. So we'll start with primary route. So you can see atrazine, it can move through runoff through water and then also through binding to sediment particles. Diuron can leach. Imidacloprid can move through vapor, kind of what each of those icons show. And then their primary route of degradation. So we've got these first two are only microbial. Indaziflam can do both photodegradation and microbial. And then bifenthrin is more specifically only when it's in soil, it can be broken down. As for bioaccumulation, we've got these three that are very much yes. I put a tiny little clam for diuron because it's like a low potential, but it still has been observed. Oh yeah, and this is my clam too. So talking really quickly about their toxicity to aquatic organisms. So the LC50 or the lethal concentration 50% is a term that is used, it's an endpoint used for regulatory purposes. And so what it means is just the concentration at which 50% of the tested population is dead. So we're going to look at fish, algae, cladocerans, which are these little water fleas, and they're the standard test organism that represents crustaceans, so like crabs and such, and shrimp. Eelgrass, bivalves, and echinoderms, which are like sea stars and urchins and all that. So, which pesticides are more toxic to each group? So bifenthrin is actually most toxic to fish. And then for algae, it is most sensitive to diuron and atrazine. Cladocerans are more sensitive to bifenthrin and imidacloprid. So if you see there's kind of a trend here, right? We've got the herbicides are more toxic to the algae, the plant, and then the insecticides. The fish is interesting, but like the crustaceans kind of make sense. You might notice that there's no indaziflam. So we don't have any information for indaziflam. And then interestingly, there are no studies with this LC50 for eelgrass, bivalves, and echinoderms. So first, before moving on, I want to talk about sublethal effects too. So there's a ton of these, but I just want to kind of go over some categories. These include behavior, development, which can be growth or internal, population, reproduction, biochemistry, cellular, genetic, their immune system, and hormones. So these might not mean immediate death. However, long-term, they are going to be important. If your immune system's not working, you're not going to be functioning very well in the environment, for example. So some major gaps in this research. Like we mentioned, we don't know, there's no LC50 information for all 5 pesticides for these 3 groups, for eelgrass, bivalves, and echinoderms. And then indaziflam has pretty much no research in general about its toxicity, which is wild to me. And then bifenthrin, interestingly, there are gaps in both algae and eelgrass, which may be because it's an insecticide, but it's still important to consider. And then bivalves and echinoderms. So the takeaways from this chapter: huge literature gap for indaziflam. We don't know very much about its toxicity. There are literature gaps in organism toxicity for eelgrass and bivalves, as well as echinoderms. These have long-lasting effects with these non-lethal effects, and they may be passed down through generations. Next steps: we need more regulatory research of this kind, so we need to know the toxicity to more of these organisms. We also should have a better understanding of what these non-lethal effects are and how they might affect populations over time. Additionally, combined effects of these contaminants, because we know one pesticide is not the only thing that's in the environment. And then considering the non-lethal effects, looking at maybe a generational. So usually with these experiments, you have an organism in a tank for a certain amount of time, but there are very limited studies that actually look at multi-generational effects, mostly because it's difficult. But so yeah, this leads me to my research on eelgrass, bivalves, and echinoderms. So this third chapter, this is looking at co-occurrence of pesticides and pharmaceuticals and personal care products across tribally important Zostra marina, which is common eelgrass communities. The first, why eelgrass? Why does it matter? They're nurseries for many important organisms. And so truly, when I say nursery, I mean like the babies come to these habitats and they're kept safe from most predators so they can grow while they're in this sensitive life stage. And this includes crabs and salmon. And then it also houses a lot of clams in those mudfloods. And these are all First Nation foods too, so they're traditionally important ecosystems. They're so important, they're actually legally protected and managed at a federal and state level for their importance and their contributions to fisheries. They're also really important carbon sinks. They actually fix carbon 30% faster than rainforests do. And in Oregon in particular, they have died off by 24% since the 1870s. And I read a paper too in the South Slough in Coos Bay, it was 40% between 2016 and 2017, which is like pretty hefty. So for this study, our main partner was with the Confederated Tribes of the Coos, Lower Umpqua, and Siuslaw Indians, who are working on their environmental contaminants lab. We also worked with the U.S. Geological Service and the South Slough National Estuarine Research Reserve. So the concerns that were brought up, especially the tribe was concerned about contaminants in bivalves, and this included not only pesticide application, because in Oregon on the watersheds, there's a lot of forestry and agriculture activity, but also wastewater effluents. And some of their gathering sites are right next to where wastewater comes out. And so this includes pharmaceutical contamination. And then the South Slough had told us about some pretty hefty eelgrass die-offs farther up in the estuary that they were concerned about. And so we wanted to look at what the cause might be and how contaminants may play a role in this. So for the study, we decided to do before and after the industrial spray season. So this encapsulated about 2 months. We looked at lower and higher impact, which had to do with the wastewater effluent. So that ended up being 4 sites in total. And we sampled multiple environmental compartments. So this included water, clams, sediment, eelgrass, and posies. And so what the heck is a posies? So polar organic chemical integrative sampler. That is why we call them posies, because nobody can say that all the time. So they're these passive samplers. So when you go out into the field and you collect a grab sample of water, you're only getting a time capsule of that one, like an image into that one moment. But if you put a passive sampler out for long term, which is about a month or so, everything that passes through the membrane, which you can see in the image there on the bottom left, is going to get collected. And so you can see what goes through there over a longer period of time. So our site locations in Southern Oregon included Florence. The impact site is outlined here in pink. And this was our lower impact site. And from now on, North Fork Siuslaw is going to be NFS just for space. And then in Coos Bay, Fossil Point was our higher impact site. And the South Slough Estuarine Reserve was our lower impact site. So to begin, we met with stakeholder groups, which included regulatory agencies and watershed councils. And this was to kind of determine the industrial spray timing, as well as which sites we should consider, in addition to the ones that the tribe was concerned about. And for the before sampling, this started in March, and this was in 2023. So we collected water, clams, sediment, and eelgrass. And then in April, we went out and deployed the posies. And for the after sampling, we went out and took back the posies and collected all the water, clams, eelgrass, etc. So our findings, this graph includes all of the media for both times. So it's just an image of everything that we collected. And these are based on counts. So we got 10 pesticides, which includes 6 herbicide, one herbicide degradate, which is essentially when a pesticide breaks down in the environment, sometimes it breaks into more pesticides essentially, which can be equally or more toxic than the parent pesticide. So that's what this degradate is. 2 insecticides, 1 fungicide. And then for the pharmaceuticals and personal care products, 3 antibiotics, 1 anti-diabetic agent, 3 estrogenic compounds, and 1 stimulant. And so we've outlined these here just for a clearer image so you can kind of see the difference between the 2 types. And there's a little asterisk there. These were only sampled in clams due to funding limitations, and so that's why there appear to be a lot less. Additionally, I want to point out that at the North Fork Science Law, there were no pharmaceuticals, which was what we were expecting, so that's pretty cool. So this is broken down into the before and after, and then all the different media types, so you can really see what we got. I want to point out that the axes are not the same, and that's just because different concentrations show up in different organisms and what have you, so you can see the bars better. So first, for the pesticides, you can see there are more detections before. So we missed the spray season, so we got like right after instead of before, but that's okay, because this is still a cool image of what happens to them over a couple of months. So interestingly, you see higher concentrations before, except the sediment. And so that one is down here. Got this graph, you can see it's much higher than in the original or in the before sampling. And that is because those contaminants actually take a little bit longer to get to those sediments. So the mobility is slower. So it kind of makes sense. There we go. And then again, in the surface water, there were none at the North Fork Science Law, which was interesting. As for pharmaceuticals, they were a lot more variable, which makes sense. There's not really a particular season for most of this stuff. There were none at Fossil Point in the before samples, and then none again at the North Fork Science Law. Okay, so this is kind of a big table. Essentially what I did, I wanted to talk about the uses for all of the pesticides. And so each of the boxes is kind of sized proportionally to how many pesticides were found from that category. So you'll see agriculture is the biggest one. Almost all of them are used in agriculture. And then we've got a bunch for commercial pesticide use, turf, infrastructure, which includes like wood preservatives and paints, ornamentals, which is like Christmas trees, plants that are more visual than edible. The residential insect management. This little one is forestry. And then we have pet flea medication and then aquaculture. And then for detected pharmaceuticals from wastewater, so this one's a little different since they're coming from one particular spot. So we have categories instead. So this is antibiotics. We have hormones, all 3 of them related to estrogen, anti-diabetic agent that I actually take myself, and stimulant, caffeine. I think that was not really surprising to anybody. It's in everything. We drink a lot of caffeine. So takeaways from this chapter. Pesticides and pharmaceuticals do co-occur in eelgrass habitats. These chemicals are in the clams and they're getting consumed by humans, and especially the tribe. And since tribes are eating them at higher rates, they're going to be taking in higher concentrations. We need to focus on the reduction of pharmaceuticals in the environment. And I want to talk about this. This isn't a reduction in use. This is more a reduction in the way they get to the environment. And so we need better disposal methods. As of right now, I don't know if any of you have tried to take your medications to a take back box, but it's pretty complicated. So we need a more available system. And currently wastewater treatment is doing its best. So hard, but unfortunately these chemicals are not able to be cleaned out during that process. So they make their way into the environment, and this involves this includes pesticides too. We also need a better understanding of uptake by eelgrass. A lot of the studies are looking at heavy metals, which are of course very important, but we don't really know what's being taken up by eelgrass. And so we were able to see some of that here, but because we didn't necessarily look at the eelgrass independently, we may have some residuals that were on top of the eelgrass, so we don't really know for sure the mechanism, but it would be important to look at moving forward. And finally, full ecosystem consideration for pesticides. I feel like when we talk about toxicity stuff, we're just talking about water exposure, but here we're seeing it's coming from a bunch of different places. And so organisms are getting exposed from multiple routes. All right, so chapter 4 This is the clam study. I didn't come up with a name. That was my friend Patrick. I got to give him credit for that. It was awesome. So yeah, silence of the clams. Forestry registered pesticides as multiple stressors on softshell clams. So just to start, forestry in Oregon is it's very prevalent. There's a lot of it taking place with watersheds, and so those contaminants will make their way into aquatic systems pretty easily. This study is building off of research by one of our former lab mates, Kagan, where he went out and categorized different land use sources as well as pesticide presence in water and softshell clams in addition to other bivalves. So I wanted to ask, what are the combined pesticide effects on softshell clams? So this is the softshell clam, my arenarium. And the pesticides that we chose from the study were atrazine, hexazinone, indaziflam, and bifenthrin. I just needed to share this picture. Yeah, like Elise said, we got like 600 something clams. So this is me and Steve. Really appreciated that. Came out beginning of COVID. And I want to point out, these are invasive clams. We did not by any means decimate the population. They're fine. And when the tide comes back in, the mudflat kind of resets itself. So we didn't really damage the area. The experimental design, we did chemicals on their own individually, and then a two-way combination, and then a multi-way. And you'll see bifenthrin only shows up once, and that was truly a space limitation. And so we just wanted to make sure we had at least one treatment where we could compare the bifenthrin with the other three. And so I dosed these every 10 days, and then they were sampled every 30 days for 90 days. Yeah, there's bifenthrin. So for measured effects, we looked at mortality, shell length, condition index, which is essentially the soft tissue over the hard shell. And we look at that because sometimes when bivalves are really struggling, they'll still grow their shells, but they might be shrinking inside. So it's a kind of an incomplete way of telling how they're doing. And then we looked at algal clearance rates to see how they were feeding. And then the water and tissue composition to see where the pesticides ended up. So first for mortality, all of them were significant. So just looking at this box and whisker plot, if folks haven't seen these before, essentially this dark line here in the middle is the average, and these show the full spread of the data points. And so you'll see down here on the left, and this is consistent for my figures, the control is kind of always off to the left, and then all the treatments are here. So these were all much higher compared to the control. And actually, the only clam that died in the control was on the very last day of the experiment, and I was very upset about it. So close. But indaziflam was the highest out of all of these. And again, this is the one, remember, there's like no toxicity studies on indaziflam. And so we're going to have this running effects table off to the right so we can make sure to see all of the things that have happened. So then looking at shell length, the atrazine hexazinone treatment had actually longer shells, which was interesting. So it seemed like the growth was doing well. As for condition index, so again, this is the soft and the hard tissue. Hexazinone and then hexazinone and indaziflam by itself were both significantly lower than the control. And then this graph is tricky, so don't stare too hard at it, but essentially this is over time. We did every few weeks for this, and so you've got 1, 2, and 3 samplings compared to the baseline start off to the left. And so you can see they kind of go up near the end, which is interesting. But the ones that were significantly different, I've circled here, they were lower than the control. And so these were the indaziflam, the atrazine hexazinone, and then the 3-way combination. And then additionally, looking at the concentrations in the water and the tissue. So here you can see when we dosed the water on the left with atrazine, there were some concentrations there, and then there were also some that were taken up in the tissues by the clam. Indaziflam was actually higher. These were dosed at the exact same concentration, so these were both 5 micrograms per liter. However, indaziflam was taken up more by the clams. So looking at all this again, we're going to go through the takeaways. Indaziflam is toxic to softshell clams. Most of the treatments resulted in mortality. There seemed to be an additive interaction with indaziflam and then atrazine and hexazinone in all 3 of them. And so an additive is essentially when you put those compounds together, all of their effects add up. Whether you could also have synergy or synergistic effects, which are like multiple, yeah. And then antagonistic, sometimes they kind of outweigh each other and you won't see any effects at all. So this is something we can't conclude with this, but there were different non-lethal effects, which may mean different modes of action. So we see for different groups, like the indaziflam and those have different feeding rates. However, they weren't significantly lower for condition index. So it means they weren't really feeding as much, but they weren't shrinking either. So it's interesting. That would be another future study to look at. And then indaziflam was taken up by the tissues, which is pretty cool. I mean, not great, but it's just. All right, so chapter 5. Oh yeah, in the clam study too, I meant to point this out. These are both published. And so I've got the citations on the page down below if anybody's interested. So this one is the effects of imidacloprid and polyester microfibers on the larval development of the endangered sunflower star. So for folks who have never heard about the sunflower star before, so Pycnopodia helianthoides. And so this is what's pictured here with me. So like this big. They're incredible. I love them. If folks haven't heard of sea star wasting syndrome, it was this devastating event that happened in 2014 where sea stars along the West Coast just were decimated. And this is a very sad picture of what it looked like for this species that kind of just melted. Their arms would crawl up and we still don't really know what caused it. And so most of these species have recovered pretty well, except for Pycnopodia. And so it's currently internationally endangered under the IUCN Red List. And then it's currently under review for national status by NOAA. And there's no research on threats from contaminants to the species. So enter WA. So I went up to Friday Harbor Laboratories, got to work with Jason Hoden in his lab. And so he's been rearing Pycnopodia to try to reintroduce them into the environment. So they have them in a lab. Very cool. And so we spawned them, which essentially means that their sperm and eggs were released into the water column, and that's how they reproduce. And so once those eggs were fertilized, we were able to raise them throughout their larval development. And so through that development, we exposed them to imidacloprid and polyester microfibers. So just to kind of go over these contaminants again, we talked a bit about imidacloprid already. It's an insecticide. Currently banned in the EU because it is so toxic to pollinators, so it's banned for outdoor use. But we still very widely use it here in the US. Its observed mechanism for larval organisms, it actually disrupts the development of their stomach as they're developing. And so we chose 10 nanograms per liter because that's an environmentally relevant concentration. Additionally, polyester microfibers. So they're one of the most commonly detected types of microplastics that are detected in the environment. And they can cause false satiation. So a lot of organisms think they're full after they've had these fibers, but there's no nutritional value there. And then they can also cause physical damage. We chose 25 fibers per liter as the concentration because it's also environmentally relevant. Also, funny enough, this was the fibers, that was the scariest thing for everybody at the lab. They were like, what are you doing in here? And it was literally a fleece that just was like, yeah, it just looked really sciencey, but I was like, I got pesticides over here too. There's for the treatments. We had the controls, did just imidacloprid by itself, and then just microfibers on their own, and then a combination. And we started out with 500 larvae per jar. Now these are microscopic, and these were in mason jars too. So for dosing and sampling, we did doses at each water change, which was every other day. And since that's a lot for pesticide dosing, I would do a stock solution. So I would make it at the beginning of a week and kind of let it degrade. However, with microfibers, they're kind of just always in the environment. They take a lot longer to break down. So we did those continuously. And so this was a 6-week larval development experiment. We looked at survivorship, length, developmental features, And then we did a settlement experiment, and there's an asterisk here because I was not successful with my larvae because it is very difficult to settle little baby Pycnopodia. But Jason had some other larvae that he was culturing alongside it, so we used his, so it's a little different. So to talk about the developmental features we looked at real quick, first is the coelom. And so if you look really close here, you'll see on the left, it's not fully fused yet. It's kind of those two little things sticking out. And then on the right, it is fused. And so this marks a developmental phase. They're moving from bipinnaria, which is what that stage is called, to brachiolaria, which is their later stage. And then additionally, we looked at these brachiolar structures. And so the two on the left essentially are what forms their body as they turn into juveniles. And then on the right, that little sucker is what helps them settle. And then the rest of that becomes their legs and everything. So they need these for settlement and metamorphosis to juveniles. So the design for the settlement experiment was just 20 larvae per jar. So this was much smaller and tiny little mason jars. This was just for 48 hours. And we wanted to compare biofilm type. So we looked at adult Pycnopodia and then also diatoms. So it was just like a layer of algae. And then we did just the pesticide for this. So control versus imidacloprid. So this is what the criteria was. They were attached. So if you can see, it hasn't quite scrunched down yet. Settling is where it's about to get down there, and then settled is fully scrunched. And so this is microscopic again. So this is a coralline algae they're on. Those aren't like two fingers pointing at each other. Like everybody asked me that. These are like smaller than a pencil point. Also, if there's no credit on these photos are mine. I meant to mention that sooner too. Results. So this is every week we took pictures. So this is what their whole larval development looks like. And vertebrates are so cool. Just gonna say that. So first, the big one was survivorship. So if you look here at week 2, the imidacloprid is significantly lower than the control. And so there was much more mortality in that treatment than any other one. What we thought this might be, we also saw this gut malformation, which was kind of a surprise. I wasn't looking for this. But at week 2, at that same time, 10% of the larvae had this contorted gut. So if you look, these guys on the right have this beautiful full gut. The red is the algae. They're just living their best life. And then this contorted gut. So they're clearly not going to make it very long if they can't eat. This also happened in week 5, which was interesting. And this was in the imidacloprid and the combination treatment. And it was much fewer larvae, but we still saw it. As for length, so the controls are off to the left here. And then all of the other ones were significantly longer, which was really interesting. And so this might be a possible stress response. We weren't able to look at this in the experiment because we just didn't have the power or like enough people really to do it. But people have observed in larvae that if they're stressed, they need more oxygen, they're breathing more, and so sometimes they grow longer. So that's something to look at in the future. And then coming back to these developmental features, so this is week 2 and 3. That's why they're so different in the height there, but there was no significant effect. So either there isn't one, or maybe we didn't have enough to look at, but that's what we found for this. So some other observations that we found a lot of the fibers just lodged in the larvae. And I mean, like we tried to pull them out with a pipette and they were stuck. And so we don't know if this killed the larvae. We weren't able to monitor them. Again, we had 500 in the jar. So, but it was an interesting finding. And then near the end, when these larvae are getting ready to settle, they start sticking to stuff and they were actually settling on the fibers. Like we weren't able to pull them off. And so this is really unfortunate because if they're going to settle on a fiber out in the ocean, in the water column, they're going to drift away from any potential food sources or fellow Pycnopodia, and they're not going to be able to grow properly. So coming back to this settlement experiment, again, remember these were different larvae than my culture. So overall, we did observe a higher preference for adult biofilm, as shown here on the left, which we kind of knew that would happen. It's a really strong settlement key for them, so that was cool that we confirmed that. We also found there is a slight difference in the pesticides with the adult biofilm. So actually there was higher settlement with imidacloprid. which is interesting. Again, we can't really say why that was. Talking with Jason in previous cultures, they've seen sometimes when the water's contaminated, the larvae will develop quicker to try to get out of the contaminated water. So it's one hypothesis, but we can't confirm them. Chapter 5, takeaways. Imidacloprid is toxic to early Pycnopodia larval development. It inhibited stomach growth, which is similar to its target organism. Microfibers may have been too long for them to ingest or affect them physically, but we did see some impacts, including the wedge pieces and the settlement response. And we also saw a consistently longer larval length in all of the treatments. Imidacloprid also supported a higher settlement than in the control. And I want to make a note, this is a first of its kind. It was a very difficult experiment. Pycnopodia is particularly difficult to culture. And so that's probably why it's the first of its kind. But this makes the case for needing more of these non-standard test organisms for toxicity testing. They may not all have the same reaction, and so it is still important to know what their response is to these contaminants. So conclusions. So we talked about a lot of stuff, so just going to come back, rehash everything. So pesticides are used in many sectors. They're lethal to fish, algae, podocerans, and we do not know as much about eelgrass, bivalves, and echinoderms with this regulatory endpoint. Chapter 3, we're finding pesticides and pharmaceuticals in eelgrass habitats. Oops. Organisms may be exposed from multiple routes, so the eelgrass, the water, the sediment, and humans are consuming these chemicals. With chapter 4, indaziflam was the most toxic to this softshell clam. Most pesticides were actually toxic in the study as well. They also induce non-lethal effects such as growth and feeding. And with chapter 5, imidacloprid is toxic to Pycnopodia during early development, and that mode of action is similar to what is seen in its target organism. Both compounds affected the length of the larvae, and both of these contaminants may be affecting sea star recovery. So moving forward, what we need for research, we need a lot more of these non-standard test species. That's partially why the bivalves and echinoderms are not well represented with toxicity. And we need to consider long-term generational effects of these chemicals, not just their one instance of toxicity. We also really need to look at the combined effects of all of these contaminants since they're all arriving in these environments together. And we want to look more at the role of contaminants in disease recovery. Since this is an ongoing thing, I was one of the only toxicologists. I mean, we were like the only ones being like, hey, maybe a contaminant's not helping. And so it's really, we need a more holistic conversation about things affecting these animals. So some things moving forward, maybe changes in management for these contaminants. So again, with pharmaceuticals, definitely not saying we should stop using them. They're life-saving medications. And so what we should instead focus on is better disposal methods and then working on wastewater technology that's able to clean them out. Using reduced pesticide use, especially near bodies of water. And then moving away from agricultural reliance on pesticides, which that's a whole nother TED Talk. But yeah, thank you all so much. That's appreciate it. Acknowledgements really quick, like even more people than on the slide, but I mean, just so many people. Thank you for everything. And I've got some fun pictures. I have tons of pictures. There's so many more, but this is like, yeah, these are the ones that I say.
So the process is, we now open it to questions, but I would go last, but at least maybe pull the. In about 10 minutes, we're gonna kick everyone out, and then Ellie will defend. Not presenting.
Yes, for folks on Zoom, if you have questions, let me know. Bria, would you want to monitor the chat for questions? Yeah. Thank you. You're the best. Okay.
Okay, I was just wondering because you mentioned that indaziflam was the most toxic to clams and that it's one of the least understood pesticides, so why did you choose imidacloprid for the Pigmentodia study?
That's a great question. When we were looking at detections, imidacloprid is really commonly detected in tidally influenced regions. And when I was researching the the Pigmentodia study, that was one of the ones that was coming up more frequently than indaziflam. And so that was why I started with that. I also, when we were considering which pesticide to use, because we had to decide between, I mean, so many, right? Indaziflam is an herbicide. And even though it was toxic to the clams, we thought maybe imidacloprid as an insecticide might have a more toxic response to the sea start. And so we wanted to maybe start with that first. Great question.
What was the target organism for imidacloprid?
Thank you. Appreciate you asking this. Thank you. I meant to mention that. So they're for sucking insects, so typically mosquitoes. So they reduce the development of the stomach because apparently that's how malaria gets contracted. So if they can't grow the stomach, they're not able to keep the infected blood in there. Sense for the target organism, but not for sea stars. Yeah, thank you. Ooh, wow. Thank you. Thank you.
My question chapter 4.
Mhm.
I want to ask what's the most challenging things for something with that aside cooking.
The most challenging. Yeah, so sorry, I haven't been repeating the questions for Zoom. So what was the most challenging part of collecting the clams for chapter 4? I think it was just doing everything within the tidal cycle. Because yeah, we needed at least 300 clams because it was such a big experiment. And so, you know, you have people coming out, we would rush down to Florence, which is 3.5 hours from Portland State. And then we would only have an hour maybe to dig as much as we could. The hardest part, they're called soft shell clams for a reason. They're like potato chips. I mean, you could probably flick them and you know, and so trying to dig them up and have them in a happy, healthy condition is so hard. And so yeah, Steve showed me how to do that. I mean, you pretty much have to find the little holes where they breathe and then put a little pipe down there and then you dig around so that you don't break them. So that was probably the hardest part. Yeah, good question.
There's some 2 questions in the chat.
Ooh, okay, let's see.
Yeah, I can read them.
Oh yeah, that'd be great.
The first one is how did the water sampler not get clogged from being in the water for a month?
That is a great question. Yeah. So how did the water sampler not get clogged from being in the water for a month? I was pretty strategic on where I put them. So it was mostly water going through there. And the big thing was the casing. So here we'll go. See if I can go back. Oh yeah, all my references. So you'll see with the sampler, this external casing here is what really helps. It keeps all of the algae and all of the critters from getting on the membranes. And so the membranes themselves are actually pretty clean. The outside was covered in, but it worked well enough. It didn't get clogged just with the currents. And I try to keep them out of the mud and places like that. So I like that question.
And then the next one is how do you all test for different pesticides in your samples? I have an idea of how to do it for genetic material, but I'm less familiar with pesticides.
Oh yeah. I'm trying to narrow it down from TED Talk. Yeah, so pesticide detections, how do we test for them? There are a number of different methods and there are 2 different primary chemical machines that you can use. So there's one called LCMS, which is liquid chromatography. And then the other one is GCMS, which is gas chromatography. And they identify different compounds. So, and hopefully this isn't over explaining, but essentially when you have a sample, you kind of pick which compounds you're looking for. So that's also why it's hard to detect everything. So you get whatever that test can identify. And then even with that, we have to train the machines ourselves. So that was part of the tricky bit with this project with the tribe. Janet from TT Clusi was literally like building this lab from scratch. And so she had to sit there and train the machine to identify stuff. And so it's as much as you can train it. And yeah, I guess more like literally you just take the water sample and run it through there. And then Additionally, with the tissues, they get freeze dried and ground up. And that's how they get. There's more to it, but yeah, hopefully that answers that. Yeah.
I was curious about you're testing specific pesticides and herbicides. But each of those, when they're actually applied, are a cocktail of chemicals. So is that something to think about for future studies, or how it might impact even as a chemical get treated differently because of the other chemicals it's associated with?
Excellent question. Yeah, so that's actually something we talked about, because when we do these toxicity studies too, so for the Zoom call, when these chemicals are applied, they're with other chemicals, typically even with just the one application method, or they'll mix them in big barrels or what have you. So for the studies, we were using this like specific powder that was pure, but there are studies that actually look at if you just would take, for example, Roundup, you can actually just take it and apply it directly, because not only if you've looked at a label for these pesticides, they'll often just say like bifenthrin 0.1%, and then it just says other ingredients. And so that's like all sorts of stuff. Like it kind of helps with the application of the pesticide. So like some of them just help them when they spray, it helps them make sure that they're evenly like distributed in the water droplets, stuff like that. And yeah, so sometimes those chemicals actually can be more toxic, or they can make the pesticide more toxic. And so for toxicity studies, absolutely. With those testing methods, it's hard because you get just the individual compounds, and like, I'm not even sure how to test for it, because like surfactants is one chemical that often goes into those, and those are also in cleaning products and like detergents and stuff. But I don't even know if you can detect that with, because we looked at like organic compounds, which are like pesticides and such. So good question. Yeah.
So have you communicated, or how have you communicated these results to the agencies that manage these chemicals? And are they receptive to that? And they think that's might result in change in how these things are applied to the environment?
Yeah, great question. So for people on Zoom, have I communicated these results to people in the agencies, and what changes might these make? So with the clam study, that one was tricky, especially because I was just a fresh little grad student, super nervous. But we did present to forestry folks, and they were very mad at me because we were saying, like, for one, like bifenthrin is registered for forestry use, but it's not actively used. So they were like, you know, take that off your list or whatever, you know, and I mean, and they were kind of critiquing pretty heavily some of the pesticides, but it was very helpful because I'm coming at it just from a scientist perspective. Like, I'm just like, oh, I hate these pesticides, but they need to use them for their practices. And so it's good to have both conversations. So it was good to have them go through and check. I don't know that my research is really going to make them change much, but the one that was exciting for the Pycnopodia study, it was timing really beautifully with that NOAA report. And so because I got to work with Jason, and like, so Fiona here was with me, she got to help do the 14-hour stints with the larvae. That one, we actually got in contact with the folks doing the NOAA report. And so those results are going to go into that. And what they basically told me is that once that gets published and Pycnopodia gets listed, that contaminant, so imidacloprid will not be able to be applied where Pycnopodia might live. Yeah, which is pretty exciting, because like in the past, imidacloprid has been proposed for a lot of like invasive and pest management. There was this whole thing in Willapa Bay for folks who are like Bellingham area. There are these things called burrowing shrimp. If you've been on a mudflat, you might have seen them, but yeah, they like destroy the infrastructure, and I love them. I think they're great. But if you're an oyster aquaculturist, you might not want them messing up your habitat. And so they actually proposed to go out and spray imidacloprid everywhere to get rid of them. But the town, like the community was like, no, this is horrible, please don't do this. And they actually didn't do it. So it has been proposed for use on the coast before, but this will definitely like solidify it. And then with microfibers, that one's still a work in progress. I mean, there's legislation in Oregon right now looking at putting the washing machine filters on washing machines, especially on the coast. They used to have them, but we got rid of them because people didn't know to change them, and capitalism and such. But yeah.
know you're right now defending your PhD and all the studies that you've done. But do you have experiments already in mind for the future, things that you wanna look at based on all the work you've done?
I have an entire notebook. I love that question. Yeah, because literally, I mean, like when you're doing an experiment, you think of like 50 more questions, like, and especially with Pycnopodia, I mean, literally, we had a moment when we found the little lodged fiber where we're like, okay, let's do a side experiment. And so we like took 5 of them and put them in different things, but then they all died because they were like, what are you doing to us? And like, you know, so yeah, you think of, I literally an entire notebook. Yeah. Yeah, I love that question. So yeah.
There's one more on Zoom.
Oh, yeah.
No, no, hold on. Would another going forward subject be to ask about the effects of these pesticide accumulations in the people of the tribe, even though it would be more complicated?
Yes. So for Zoom, yeah, the question, I guess you guys can read it, but would a study going forward be to look at the contaminants in the people of the tribe? I mean, it would be interesting. I'd be curious if you could do it kind of like with PFAS. I don't know, with those back when that was becoming a whole thing. It's been a 20-year case, but they were actually going through and testing the blood of people in communities to see how much they had in their blood, in their bodies. It'd be tricky with pesticides because some pass through so quickly, but with pharmaceuticals are designed to just pass through the body. They're really water soluble, so I don't know that they would stay. It would be tricky, but that is a great question. I mean, it would be interesting to see how those contaminants are moving up the chain. And so I think the best way would be to probably mimic it with a different organism that we could look at. But yeah, I like that question.
So, we're actually gonna be doing it upstairs in 20.
Yeah.
Yeah, so we'll take like a 5-minute break. For the committee.
Let me stop sharing. Okay, everybody who's left on Zoom, you're amazing. Thank you for coming.